Hello everyone, welcome to BeBiological. Today we will discuss about tools of recombinant DNA technology. Three types of biological tools are used in the formation of recombinant DNA. These are enzymes, cloning vectors known as vehicle DNA, and competent host for transformation with recombinant DNA. In this video, we will only discuss about enzymes. The enzymes used in the recombinant DNA technology fall into four broad categories. These are nuclease, template-dependent DNA polymerase, end modification enzymes, and ligases. Nucleases are the enzymes which degrade DNA molecules by breaking the phosphodiester bonds that link one mononucleotide to the next. There are three different kinds of nuclease. One, exonuclease, which remove nucleotides one at a time from the end of a DNA molecule. Next, endonuclease are able to break internal phosphodiester bonds within a DNA molecule. Last, restriction endonuclease, which is also part of endonuclease, is a bacterial enzyme that cuts double-stranded DNA by cleaving the phosphodiester bonds at specific nucleotide sequences known as restriction sites. The term restriction comes from the fact that these enzymes restrict the entry of foreign DNA in the bacteria. Restriction enzymes, therefore, are believed to be evolved by bacteria to resist viral attacks. The name of any restriction endonuclease consists of three parts. Here, I will explain it with an example of EcoR1 restriction enzyme. An abbreviation of the genus and species of the organism to 3 letters is the first point. Here, Escherichia coli is the name of the organism, Escherichia is the name of the genus, and coli is the name of the species. We are taking eco, E, from the genus name and CO from the first 2 letters of the species name. Next, a letter, number or combination of the two to indicate the strain of the relevant species. Here, RY13 is the strain of the Escherichia coli species. We are taking only the first letter, that is R. Now, a Roman numerical to indicate the order in which different restriction modification systems were found in the same organism or strain. Here, 1 is the first identified or the order of identification in the bacterium. Restriction enzymes have certain unique features which make them indispensable in recombinant DNA technology and biotechnology. These characteristics are: the restriction site sequences recognized by most restriction enzymes are palindromes, which are explained as the sequence of bases in the same on both strands when read in the 5' to 3' direction as a result of a two-fold axis of symmetry. Next, most restriction enzymes are highly specialized to such an extent that they recognize only a single recognition site or restriction site. Next, the restriction site is recognized irrespective of the DNA source. Rather than cutting DNA indiscriminately, a restriction enzyme cuts only double helical segments that contain a particular nucleotide sequence, known as a recognition sequence, always at the same site. The recognition sequence for the vast majority of restriction endonucleases are normally palindromes. Restriction enzymes make either blunt or staggered cuts. Thus, restriction fragments may have blunt ends where the cleavage points occur exactly on the axis of symmetry. Here, the example is shown by the EcoRV. And next, the overhanging end or the cohesive termini or sticky end. The cleavage point do not fall on the symmetry axis, so that the resulting restriction fragment possess so-called 5' overhangs or 3' overhangs. Here the example is clearly shown with EcoR1, where a 5' and a 3' staggered ends are clearly shown in the picture. Restriction endonucleases are categorized into three general groups: type 1, 2, and 3. Comparative analysis of these three types of restriction endonuclease are shown in the following table. The nature of enzyme in type 1 and 3 both are bifunctional with both endonuclease and methylase activity, whereas type 2 enzyme is unifunctional with separate endonuclease and methylase activity. This unifunctional activity make it unique. Now second is protein structure. Type 1 is made up of 3 different subunits, whereas type 3 is made up of 2 different subunits, while type 2 have 2 identical subunits, which make it simple. Now in case of restriction requirement, type 1 require ATP Mg2 plus S adenosyl methionine. Type 3 required ATP and Mg2 plus, while type 2 only required Mg2 plus. Cleavage site, only type 2 can cleave at or near restriction site. That is why it is mostly used in our DNA recombinant technology, while type 1 cleaves at random site up to 1000 base pair away from restriction site, while type 3 cuts near 3' to 24 to 26 bb of the restriction site. Coming to the example, common example of type 2 is EcoR1, type 1 is EcoB, type 3 is EcoPI. After nuclease enzyme, the next important enzyme is template-dependent DNA polymerase. DNA polymerase enzymes that synthesize new polynucleotides complementary to an existing DNA or RNA template. Different types of DNA polymerase used in gene manipulation are DNA polymerase 1 or Kornberg polymerase, which has both the 5' to 3' and 3' to 5' exonuclease activities and 5' to 3' polymerase activity. Now next is reverse transcriptase, also known as RNA-directed DNA polymerase, synthesizes DNA from RNA. Reverse transcriptase was discovered by Howard Temin at the University of Wisconsin and independently by David Baltimore at about the same time. The two shared the 1975 Nobel Prize in Physiology or Medicine. Now next is Taq DNA polymerase. DNA polymerase derived from a thermostable bacterium, Thermus aquaticus, is known as Taq DNA polymerase, which operates at 72 degrees centigrade and is reasonably stable above 90 degrees centigrade and used in PCR. It has a 5' to 3' polymerase activity and a 5' to 3' exonuclease activity, but it lacks a 3' to 5' exonuclease, that is the proofreading activity. Now next one is end modification enzyme. End modification enzymes make changes to the ends of DNA molecules. First example is terminal deoxynucleotidyl transferase. This enzyme is used for the formation of a cohesive end by homopolymer tailing. A homopolymer is simply a polymer in which all the subunits are the same. Next, alkaline phosphatase, obtained from various sources including E. coli and calf intestinal tissue, removes phosphate groups from the 5' ends of DNA molecules, which prevents these molecules from being ligated to one another. Next is T4 polynucleotide kinase, obtained from E. coli cells infected with T4 phage, performs the reverse reaction to alkaline phosphatase, adding phosphates to 5' ends. Its main application is in the end labeling of DNA molecules. And the last important enzyme used in recombinant DNA technology is ligase. DNA ligases join DNA molecules together by synthesizing phosphodiester bonds between nucleotides at the ends of two different molecules or at the two ends of a single molecule. DNA ligases commonly used in cloning experiments are those obtained from E. coli or from the bacteriophage T4. This is all about the enzyme used in recombinant DNA technology. Thank you very much for being with me. Hope this video helped you. Please subscribe to my channel and share the video if it helped you. Wish you a great future ahead. Thank you.